Pokud mě neklame pohled na hodinky, tak ukazují 12. A to je přesně čas, ve kterém máme začít další přednáškový blok této mezinárodní vědecké konference. Věřím, že jste se občerstvili a máte další energii s námi zůstat i v následujících minutách. Tak pokud mě ještě někdo slyší mimo přednáškový sál, pojďte prosím dovnitř, abychom mohli začít. Bezpečnost a udržitelnost zemědělsko-potravinářského odvětví, to je další téma aktuálního bloku Mezinárodní vědecké konference s názvem European Agri-Food Sustainability and Innovation. Příspěvky se v těch následujících minutách budou týkat především bezpečnostních aspektů užití nových genomických technik a také jejich potenciální role v zabezpečení evropské produkce potravin. Ostatně už jsme se tohoto tématu lehce dotkli i v tom dopoledním bloku. Dámy a pánové, dovolte mi, abych vám jenom velmi stručně představila prvního přednášejícího. Kdybych měla přečíst celé jeho kurikulum vitae, tak si myslím, že bychom tady seděli minimálně 1,5 hodiny. Takže jenom stručně. Pan profesor Jaroslav Doležel je vedoucím výzkumným pracovníkem Centra pro strukturní a biologickou analýzu rostlin a funkční genomiky Ústavu experimentální botaniky v Olomouci a je také profesorem molekulární biologie a genetiky na Fakultě životního prostředí Univerzity Palackého v Olomouci. Jeho výzkum se zaměřuje na organizaci, funkci a evoluci rostlinných genomů. Mimo jiné vyvinul metody chromozómové genomiky, které usnadňují sekvenování genomu a klonování genů u zemědělských plodin se složitými genomy. Je také velkým zastáncem přenosu molekulárních metod do šlechtitelské praxe a je koordinátorem jednoho z programů Evropské komise pro strategie AV21 Akademie věd České republiky s názvem Potraviny pro budoucnost. Za své vědecké výsledky získal pan profesor nejvyšší a nejprestižnější české národní ocenění v oblasti vědy a výzkumu Česká hlava, který pouze jednou ročně uděluje vláda České republiky. Dámy a pánové, dovolte mi tedy, abych sem pozvala pana profesora Jaroslava Doležela z Ústavu experimentální botaniky Akademie věd České republiky. Pane profesore, prostor je váš.
Moc děkuji za představení. Thank you very much for the introduction. It's a great privilege and honor for me to speak today. As you see from the title of my talk, I'd like to discuss the new breeding techniques, and even if we heard about that it may not be necessary to reconvince ourselves about these techniques, I still believe, and I agree with a member of European Parliament, Mrs. Šojdrová, that we need to continue with this activity, because after all, the politicians decide, and the politicians need to be elected, and they will follow the crowd. Excuse me this expression. So we need to convince the public about the need and necessity of the new techniques and also about the safety. So that's my talk. But I will start more generally, why we need to discuss these techniques. The simple answer is simple. We simply need to produce more food. The food security is undermined. We know that globally over 800 million people suffer from lack of food. Over 2 billion people do not have enough quality food. And the situation may only get worse because the global population keeps on growing, keeps on increasing. We are already 8 billion. We will be 2 billion more in just a few decades by 2050. And not only the population growth is the problem, the climate change, the COVID epidemics, pandemics, and also the Russian invasion to Ukraine. So we have a complex issue, and obviously we already heard today, there is no simple solution to a complex problem. So, but one way or another, one of the components of solving this problem, or one of the answers, is to produce more food. And because most of our food comes from plants, even our animals and farm animals are fed by animals, we need to increase the production of agriculture crops. And that's my talk today. However, this is a great challenge. How we can increase the production of agriculture crops? Obviously, by increasing the yields. But it seems that the yearly increase in yield is slower than what is needed. At the same time, we produce the amount of food we are producing. We produce only thanks to advanced cultivars, the elite cultivars, but also intensive agriculture. Our crops need a lot of water, our crops need fertilizers and pesticides and fungicides and herbicides. So the agriculture, one way or another, is damaging our environment. We may think of increasing the land which is cultivated, but this is almost impossible. First of all, the acreage that is available to feed one person in the earth is decreasing. Why it was about 0.4 hectares in 60s, it is only 0.2 hectares now. So we have less and less land to produce the food. Somebody calculated that at current rate of production, by 2050, we would need over 600 million hectares more land to produce enough food for the growing population under the climate change. The climate change is a real problem, and there was recently the conference on climate change, and it seems that we are already getting used to the fact that we will reach 1.5 Celsius degrees increase. But if this happens, and it's almost highly probable, if this happens, the yield of global yield of crops will decrease, at least crops that are very important like wheat and maize. So the decrease may be in the range of 5 to 10%, if not more. So we have a problem. Now, on the top of it, and rightly so, European Union is motivated to protect the environment, and so we have the European Green Deal targets for 2030, and we already heard about that. And the ambition to decrease the quantity of fertilizers, the agrochemicals, and also perhaps promote the environmental friendly agriculture, if you wish. This is maybe a good ambition, but it goes against the need to increase the production. And I attended a conference in Prague on a similar topics like 1 month ago, and I really like to thank the colleagues from the Euroseeds organization and Petra Juraš, who was there, who reminded us that if we accept these measures, the decrease of production may reach up to 20 or over 20%. So we need to increase the production, and we don't have a clear idea how to do it. And obviously there are several different measures like reducing the food waste, precision agriculture, etc., etc. I want to talk, and I want to promote and support the idea of developing new generation of crops of varieties that are adapted to climate change, and that will still provide, or they will have stable and high yields. Now, how we can do that? This is a real problem, because in reality we dream about a new generation of crops that have completely new properties, new characteristics. And if you imagine a list of changes or traits that you like to have, well, we want to increase the yield under stress conditions. So the plant should provide and produce enough yield under the drought or flooding or either excess of water or lack of water and chilling and freezing and even salinity, because we have more and more land which is contaminated by salt. At the same time, we already heard today about the effect of climate change on the invasion of new pathogens and new insect organisms that may again cause decrease in yields. Now, if we want crops that can produce enough yield under these conditions, we really need to change the properties of these plants dramatically. We need to probably change the body plan. We need to plan the organization of plant tissues and organs. And this is such a huge change that can be compared only to domestication. Only during the domestication, the wild progenies of our crops were changed so much that our crops don't look like the progenies. And I know that this is an ambition plan and it is ambition vision, but it happens once, and hopefully it may happen again. I probably don't have to remind you about what happened during the domestication and remind you of the changes between the progenitors of our crops and our cultivated crops. And you have a list on the screen. The photoperiod, change of photoperiod, daylight independence, vernalization, dormancy, determinate growth, shorter length, etc., etc., etc. So we want to change our crops, and the question is how to do that. And to answer this question, we should answer the question, what happened during the domestication? Because this was the same process, or the same type of the process. And we know the answer. The answer is change in genetic information. Because the plant, the growth of the plant, the development of the plant is regulated by the genetic makeup of the plant, and obviously with the interaction with the environment. Now, during the domestication was a process that took thousands of years. So we simply cannot sit down and wait for 1,000 years when hopefully the new types of plants will appear, and we will propagate them and use them. So we don't have time, and we cannot repeat the same. We cannot employ the same strategy or the same. We cannot repeat the same situation which concerns the domestication. Yes, we may consider inducing more changes in DNA by irradiating or chemical mutagenesis, but we all know how inefficient this system is, this approach is. There are many mutations that are induced across the genome. We don't know where they are. And if we know what to change, there's a little hope that a simple experiment will result in the plant that you may need. So what we can do, we can still, I want to argue that we can still follow the nature. We like to have our crops new genes that they are missing. Either they lost them after domestication and during breeding, or they never had them. And if you look at the nature, you will see that the borrowing the genes, transferring genes from one organism to another is very common. We call it horizontal or lateral gene transfer. So this is a natural mechanism during the evolution, and it was a very crucial evolutionary mechanism that led to development of new type of organisms. In fact, a recent study identified at least 100 gene families, not genes, gene families that were transferred to plants and that were crucial for development of land plants. So our plants evolved thanks to the transfer of genes, and the majority of donors of these genes were bacteria or fungi. So this is a very, very natural mechanism. I like very much to remind a study in relation to the opposition to Agrobacterium tumefaciens transformation. A recent study identified over almost 400 plants or species out of 475 that contain DNA from Agrobacterium. It was not introduced in a laboratory. This happened naturally. So in the nature, we see the transfer of genes from one organism to another organism, and we even see transfer of genes from Agrobacterium to plants. So that's a very natural organism. And yes, we can use this system, and has been used. This system has been used to transfer genes from different organisms to plants, to transfer beneficial genes that improve the ability of our crops. And I will not go into the details into this. There's another mechanism which is now gaining a lot of attention that can help us to modify genetic information in a very rational way, in which we can make literally a surgery of a genome. And I like to call it a self-navigating scalpel. And again, that was developed by the nature. That's not a creature of somebody who wants to harm the humankind. This is a system that evolved as adaptive immune system in bacteria, in Archaebacteria and bacteria. And in this system, as you probably all know, the bacteria will take a sample of an invader, of a harmful organism. For bacteria, it may be a virus. And will remember, or will save that piece of information to remember the invader. And once the bacteria is invaded again by the same organism, it will destroy it. It will send this scalpel to cut the DNA, the genetic information of that organism. We can use this system to make a surgery of a genome of our plants, and Professor Pečinka, who will talk in this session, will give you more information on the system, and I will again not go into the details on that. We talk about new breeding techniques, and in fact, these new breeding techniques, which we hope can bring in a shorter or sooner or later, can bring the improved crops. And these breeding techniques, the cornerstone of these techniques is the modification of the genetic information of the plants. And we can either consider genetic transformation, and again, I will not discuss the technologies, the transgenesis, where we transfer gene from one organism to another organism, cisgenesis within the similar plants, relatives, or even infragenesis, where we change the regulation system. And once again, we do nothing else than the nature has been doing since ever. Genome editing, gene editing is the system which is based on the second approach, or the second mechanism I introduced to you, this adaptive immune system of bacteria. And it helps us to modify the gene. The idea is very clever, and it utilizes the ability of our genome, even our own genome, to repair the DNA breaks. So the scalpel will cut the DNA, and then our repair system, or the system of any other plant, will respond, because this is not sustainable. So either it will link it again, these to broken ends, it will fuse them together, but the repair systems are known to make errors or mistakes, and this is how we can switch off a gene that we like to switch off. By cutting it and incorrectly linking back these two loose ends, the gene will not work, and we switch off the gene and we did nothing else. And we can also provide a template according to which the break will be repaired, and in this way we can introduce changes. But Professor Pečinka will talk about it more, and I will not go on to that. So my argument and my talk, it's a brief talk, so I cannot cover the whole spectrum of problems. My vision, or my opinion, is that talking about new breeding techniques, which are based either on gene transfer or gene editing, we are not introducing an unnatural mechanism. We are simply leveraging something that evolved during the evolution, during the thousands or millions of years of evolution, the mechanisms that were principal mechanisms for the evolution of the diversity that we see, and we simply like to use it to improve the crops. And just to provide some more arguments. So in the next two slides, I just want to remind you how successful these approaches are. So talking about gene transfer, genetic transformation, which is not very popular, but the reasons are really not very rational. It's successfully used and the crops are grown on over 4% of global land. It's not a small piece of land where the genetically modified crops are grown. More than 500 varieties were already developed using this technology. And the arguments are clear. We know which gene was introduced, we know where the gene in the genome is, and we can check the function. We can follow the transcription and protein synthesized based on this DNA. So we should not be worried. We can test. And if by any chance the proteins or regulatory molecules may be harmful to human, this plant is simply autoclaved and destroyed. So this is so easy to work with plants concerning this technology. And again, I list only a few advantages. We have GM crops that have increased yield, improved quality, new properties. It is estimated that the use of these crops reduce the agrochemical use globally almost by 10%. So European Green Deal. But this happens without European Green Deal, simply by applying and using GM crops. And obviously it leads to higher profit of farmers. For the CRISPR-Cas, and again, Professor Pečinka will talk about them more. Despite the short time between the discovery of the system, development or modifying the system so that it can be used to edit the crops, we have again over 500, probably already 600 applications of the CRISPR-Cas system. And again, only few examples. Wheat, which is not prone to pre-harvest sprouting. Wheat, which is resistant to disease. Wheat with reduced immunogenic gluten proteins. Not listed on my slide. Wheat, which is more resistant to drought, relatively recently released in Argentina. So we have a growing list of plants that use gene editing to improve the quality and yield. And I put only wheat just to make a little bit homogeneous, but I couldn't resist to present the example of tomato, which contains provitamin D. Perhaps the last slide, if I still have 1 or 2 minutes, we are talking about improving our crops, but we may also mimic the domestication and forget about the current crops, go back to our crop progenitors, or even choose other wild species with some good properties like salinity tolerance, and change their properties so that they will fulfill our requirements for cultivated crops. And once again, a nice example is de novo or high-speed domestication of tomato, when the research team used the wild progenitor of tomato, and by modifying 5 genes only, they obtained a plant which looks like a domesticated tomato, and they made it in a very short time. So to conclude, I believe that achieving food security in a sustainable manner will require development of a new generation of crops and varieties. There's no doubt about that, because our crops are not adapted to the changes, and also not adapted to agriculture that should cause less harm to the environment. We need to modify genetic information. This is clear, and the extent of this modification can be compared only to domestication. We don't talk about the gradual improvement of crops. We need a new generation of crops. And obviously the most promising strategy is to leverage natural molecular mechanisms, so that we don't have to worry, because we are using something that has been here since ever. And a good example is genome editing. And well, to conclude, my final statement is that nature has given us tools to facilitate development of such crops that will provide high yield under sustainable sustainable conditions. And I think this will. And thank you very much for your attention, and I will be happy to answer questions.
Dámy a pánové, já děkuji panu profesorovi. My v tuto chvíli nemáme bohužel prostor pro otázky a odpovědi, ale přesto, kdyby měl někdo z publika dotaz na pana profesora, nevadí, pokud nemáte, pan profesor s námi ještě zůstává v následujících minutách a samozřejmě individuálně vám potom zodpoví veškeré otázky. Děkuji, pane profesore. Ještě jednou, dámy a pánové, děkuji. Mě tam zaujalo několik informací pro mě naprosto samozřejmě nových, objevných ve vašem příspěvku. Taková spontánní mutace rostlin a jejich následná adaptace na současné podmínky by zabrala tisíce let, tak samozřejmě my jsme o mnoho dál a máme nejrůznější metody, jak udržet zemědělství v dobré kondici, tak uvidíme, jestli se to vědě dál podaří. Dámy a pánové, dovolte mi, abych teď pozvala, i když jenom virtuálně, našeho dalšího hosta, EFSA opinions on new genomic techniques. That's how Anna Afonso from the European Food Safety Authority called her contribution. Dr. Afonso, I hope we are connected. Can we hear each other?
Yes, yes, I can hear very well. Can you hear me?
Perfect. Well, so welcome to today's conference. And at first, allow me to briefly introduce Dr. Afonso. She is the head of the Emerging Risks team at the European Food Safety Authority. Anna Afonso specializes in veterinary studies in aquatic animals and joined EFSA 22 years ago as a scientist in the Animal Health and Welfare Department. Anna Afonso worked as a research assistant at the Portuguese Veterinary Faculty in Lisbon and also in Villarreal as well. So, Dr. Afonso, the floor is yours and you can start your contribution.
Okay, so first of all, let me check. Can you see my screen because I'm sharing my presentation?
No, we don't have it, so we can't see it.
Let me see if because I did share screen. Okay, so let me see if I can. What about now?
Nothing. We just can see you.
Okay, what about now?
Nothing, but you look great.
That's very kind, but that was not the idea. Is it taking a little bit longer?
We've got it. European Food Safety Authority. Yeah, the floor is yours. You can start.
So, first of all, let me make a bit of a correction here because I think I'm not quite sure what happened with my biography, with what I provided and what you read are not exactly the same thing, which is not that relevant. I'm still the same person, Anna Afonso, but I now lead the Nutrition and Innovation in Food unit and no longer the team on emerging risks. So, and it's in this role that I'm giving this presentation. First of all, I would like to thank very much the previous speaker. Very inspiring talk. Thank you so much. And I think you covered some of the things that I would like to say as well. So that will save us some time. So let me, whoops, this is going or not? Yeah. I think it's nothing new for this audience of what we do at the European Food Safety Authority. So our mission is to ensure food and feed safety, but also protecting the environment. And within the scope of this conference, I'm going to focus, of course, on GMO. Risk assessment of GMO plants in the European Union. The slide is not so good because it's rather small, but you can find the same infographic in our website. But it basically summarizes what we do in terms of risk assessment. When we're talking about risk assessment of GMO plants, we're always talking about comparisons. So we compare the genetically modified plant with a conventionally bred plant, so something which has not been genetically modified. Very important. the assessment is always on a case by case. So there's no generic risk assessment. Each product which is notified to be imported or cultivated in the European Union goes through this process where the sequence of steps on risk assessment are followed to ensure its safety. And these steps are, first of all, molecular characterization. How was it created and what are the changes? A comparative assessment where we look at the composition of the products of this plant, a phenotypic characterization and its agronomic properties. After these two steps are concluded, we also conduct, or in simultaneous, a safety assessment. So we look at toxicological properties, allergenicity potential, nutritional value, and last but not the least, also the safety assessment for the environment, for the receiving environment. As you also know, in the European Union member states, there are only 2 countries which are now producing genetically modified crops, and in fact only 1 crop is actually licensed at the current, well, 2, 1 on controlled conditions. What I would and I made this very brief introduction because I think it's important that we do not forget that the European Court of Justice in 2018 very clearly stated that organisms obtained by mutagenesis are GMO and are in principle subject to the obligation laid down by the Directive 2001/18, which regulates the process of assessment and authorization of such products. So, but here we start having a little bit of issues in the sense that the way these new techniques are developed brings us some challenges. So GMO are organisms in which the genetic material, DNA, has been altered in a way that does not occur naturally by mating and or natural recombination. After the previous speaker, I wouldn't go further, but there is still a human intervention, and therefore, according to the European Court of Justice this deliberation, we have to consider these new products as GMOs, no matter what they are producing. What is also being a challenge for risk assessment and for risk managers on this process is the quick evolution. When we're talking about CRISPR-Cas, we're talking about techniques that have been or others. We're talking about techniques that have been developed in the latest 12 years. The evolution in this field of science has been enormous at a speed that it's difficult for the regulatory framework to follow, and therefore some of the challenges. Now, I would also like to say that or actually repeat what I said before, that the risk assessment performed by EFSA and its panels are never limited to techniques, but consider the product as a whole, the GMO and derived food and food products. I like very much the expression genome surgery, and I will try and use it on my next presentations because I think it's a very nice way of describing. Now, Professor Doležel also talked about the differences between established and new genomic techniques, but also about the products that we are aiming to achieve. And indeed, what we are still using established genome techniques, there has been an evolution on what kind of traits are being modified, about the complexity of the traits that are being modified, about combinations of traits. New genomic techniques are very much giving us promises about increasing targets, specification and precision on these modifications, but also a range of new traits that could have a very high potential tailored to consumer needs. And the professor before just mentioned the tomato, which is able to give a precursor of vitamin D3. So these are all things which are very, very promising. And of course, as a risk assessment body, we've been working on these topics for now 10 years. I'm also not going to go into what's the difference between established and new genomic techniques, because my understanding is that future other speakers will go into more details about the various techniques. But what I wanted to highlight is that the two kinds of techniques, established genomic techniques, meaning anything which was prior to 2001, and the new genomic techniques can lead to outputs which are either of transgenic nature, cisgenic or intragenic nature. And we now have a new group where we're looking at synthetic biology, where the combinations are of a much higher complexity, but where by design we're producing a final product that accumulates a series, not just technology, but the traits are coming forward. And I would leave it at here, but I think it's important to understand a bit of our more recent work today, talk about that. So as I said, EFSA has been working for now 10 years on biotechnology mandates, and we call it biotechnology because it includes also the mandates on synthetic biology. And we've been asked by the European Commission, by DG Santé, to have more or less the same questions. And the questions are, can we identify any potential risks in terms of the impact on human, animal health, or the environment from the application of these techniques? Are there novel hazards, and here compared to either conventional breeding or established genomic techniques? And more important, are our risk assessment guidelines, the way we assess these products, still adequate and sufficient to perform the risk assessment and ensure the safety of the consumers, or do we need to update them? So the list is getting quite long. So as I said, from 2012, when we first issued as EFSA GMO panel opinion on cisgenesis and intragenesis, we've been working until very recently, and we still have more to go. I'm going to try and focus on the last two opinions, which are the safety assessment of plants developed through cisgenesis and intragenesis, which was just published in October, and an EFSA statement that has been also published in October, where we're looking at criteria for risk assessment of plants developed through targeted mutagenesis and cisgenesis, and only those two outcomes. So maybe it's worth it to give a sort of overview of our past work. So regarding those questions that I've mentioned before, and for the three opinions that you see listed in the slide, the EFSA conclusions is that there is no new hazards associated with plants produced by, and you have the list in the slide. We also said that the current EFSA guidance documents, which are aligned with the regulation for risk assessment, are applicable. But in the case of plants produced by cisgenesis or intragenesis and SDN3, site-directed nuclease type 3, they are in line, and they're partially applicable in case of plants produced by site-directed nuclease 1 and 2, and also ODM techniques. And what we've also stated in the opinions was that there is a need for a flexibility in the data requirements for the risk assessment, as on a case-by-case, lesser amounts of data might be needed for the risk assessment. So no new, for example, regarding molecular characterization, if there is no genomic alteration, etc. Another piece of work that was conducted at EFSA was just an overview of the work that has been made across European member states, also looking at new genomic techniques. And we try to keep this overview updated because, of course, we're very much interested in the work that is conducted in member states. And so here is a plea. If you're working in this domain, keep us in posted, because we're very much interested to listen to the work that you've been doing and progressing together. So this is the recent cisgenesis and trigenesis opinion of 2022, and the question was, is there something new? But there was also a slight change on the definition that was used in 2022 to make these differences between cisgenesis and intragenesis more clear. But overall, the conclusions remain valid, saying the hazards arriving are similar to those from conventional plant breeding, whereas additional hazards may arise from intergenic plants. And why are we talking may arise from intergenic plants? What was explained already is that there's, and I think I go back to my explanation on technology and end result, is that on the intragenic plants, we are inserting the same, the GNA from the same plant or a sexually bred cross plant. However, we are creating a modification on this particular genome. So, as other conclusions, the development of this kind of plants through new genomic techniques we said that it could minimize the hazards related to the introduced DNA and trait. We're not introducing a transgenic DNA. We could also minimize the potential alterations to the host genome observed because here we have techniques which are now available, which take a much higher precision. And as a final, we also stated the same conclusion regarding the guidelines that we're using, that stating that the present guidelines could be considered sufficient, and they're partially applicable for the assessment. And we say are partially applicable because, again, there, as on a case-by-case basis, some requirements may not be needed for the risk assessment. So, and there is a plea for more flexibility on the assessment of these products. Now, talking about criteria, it's well known that the European Commission, together with the member states and the Council, are working on a possible, at the moment, the European Commission is working on a proposal for a review of the regulatory framework on the assessment on these plants. This work is being conducted together with a working group of member states on new genomic techniques, but EFSA was also requested to provide a statement and advice for consideration by the European Commission on possible criteria for the risk assessment of these plants. And criteria meaning not data requirements, we're not there yet, but elements that should be taken into account to evaluate on a case-by-case basis, as it is requested by our Mother Directive 2001. And this could also lead to a future development of data requirements, what type of amount of data would be necessary. What we came out with in a very short amount of time was something that took into account all the considerations and conclusions that had been reached over these last 10 years. And together with our experts, we proposed what we like to call basic criteria, and organized in a decision tree. There's work to be done. This work was done for what EFSA standards is a very short period of time, from May to October of this year. And I think probably most of you saw this simple decision tree. But so what are those criteria? One quite obvious: is there any exogenous DNA present in this product that we are assessing? And that's criteria one. Is this DNA sequences from the breeder's gene pool? A second criteria. Third, what is the kind of type of integration that we are looking at? And four, is there an unintended interruption of an endogenous gene? So the second part of, still the same drawing. Once we follow all these criteria, we would reach to what we call criteria 5 and criteria 6. And criteria 5 talks about history of use, a concept which is not new, and not new on the European regulatory framework, not new for a lot of you for sure. So we made a slight distinction between what we call history of safe use and familiarity. On history of safe use, we're talking much more about all the considerations we do in risk assessment when we look at the product as a food and feed. So the safety of this product for the use as food and feed. While on familiarity, we're really talking more about the environmental assessment, and here we're combining between the traits and the receiving environment. And finally, as a sixth criteria, the function and structure associated to the new allele. Now, the EFSA also provided a few additional considerations that would require further work, further investments, as that we need to make sure that multiple genetic modifications can be introduced simultaneously. And so we will see products that they will combine different possibilities, and that we need to ensure that the principle of single first assessment is still maintained. In case multiple plants endogenous genes are modified to produce many novel proteins, the assessment will be challenged according to the regulatory framework. The EFSA also concluded that comparative analysis may not always be applicable to some genome edited plants with complex strains for which a comparator cannot be identified. So these are all aspects, and of course we will need to work further on these concepts on history of safe use and familiarity, on how this will be implemented in practice, since they are not directly related with our experience so far. And I think this was what I had to say. I would also like to take the opportunity to invite you all to participate on a stakeholder event that EFSA will hold online on the 12th of December. And what we intend with this event is to discuss openly with the various stakeholders what are the challenges for the future for the assessments of the safety of plants derived from new genomic techniques. Again, thank you very much. I apologize, we don't think we have time for questions, but feel free to contact me or EFSA at any time with any questions. And thank you once again for the invitation.
Many thanks. You don't have to apologize. And Afonso from EFSA has to say goodbye to us this time, as her work commitments unfortunately do not allow her to stay with us during the following presentations and program. But thank you for your time. Mrs.
Thank you very
Afonso,
much.
thank you, and thanks for your contribution to this part of the international conference. Thank you.
Thank you so much.
Dámy a pánové, a my pokračujeme, abychom nenabrali příliš velký časový skluz, i když už to vypadá, že budeme mít nějaké menší časové zpoždění. Konfederace výrobců potravin a nápojů v Evropské unii Food Drink Europe se snaží podporovat nápady, inovace a politiky, které umožňují potravinářskému a nápojovému průmyslu vyrábět produkty, které jsou bezpečné a také přispívají k zelenější planetě a zdravějšímu životu i prosperující ekonomice. Já jsem moc ráda, že pozvání na dnešní konferenci přijal generální ředitel Food Drink Europe Dirk Jacobs. Dobrý den. Let me welcome you here and briefly introducing. Dirk Jacobs je členem nejrůznějších platform Evropské unie, pravidelně se zapojuje do činnosti OSN, WHO nebo OECD, je také předsedou pracovní skupiny, která vedla vypracování kodexu chování Evropské unie pro odpovědné podnikání v potravinářství, což je také první výsledek strategie Farm to Fork. Tak, the floor is yours.
Thank you very much, ladies and gentlemen. It's a pleasure to be here, and I first want to thank the Czech presidency of the EU for inviting me, but also for your leadership in driving a very busy, complicated policy agenda over the last couple of months, and only 1 month to go, and we're there, so well done. My name is Dirk Jacobs, and if the slides are up, I will be talking about unlocking the innovation potential from a food and drink industry point of view. So you have heard a lot about farmers, you have heard a lot about very technical experts about NGTs, but I want to take the downstream perspective on what is the industry thinking about innovation here. First, a bit about Food Drink Europe. We're the organization for the manufacturing sector, so we are in between the farm and the fork. So that plays an important responsibility on us, and we bring all of these different interests in the food industry together in Brussels, and we are existing now 40 years. By that time, we have been able to become an important stakeholder for the EU and international organizations. Very briefly, our members are national food and drink federations, sectors, including dairy, meat, snacks, coffee, beer, what have you, and also large multinational companies, and we bring all of these different interests together. Now, looking at the food and drink industry in Europe, we are the largest manufacturing sector of Europe in terms of turnover, value added and employment. So this is also an important factor which is sometimes forgotten. Europe is strong when it comes to food and food production, and we need to cherish that. We're in fact also the largest exporter of food and drink in the world. So that makes us an important player also on the global stage. And with that, of course, comes an important responsibility. Now, what we are in today, we have heard it already, we've been faced with the COVID pandemic. It's not over for our industry. We're still dealing with the aftermath of this, but also the supply problems that we're facing in China, for instance. And this is having an impact. And our small companies are only now feeling the impact of the pandemic. So this is not to be underestimated. Of course, the war in Ukraine, but also the tensions that are there between the US and China, other geopolitical tensions have made us more aware of the political importance for agri-food supply chains. And then, of course, the energy crisis. Let's not forget that energy before the COVID pandemic made up some 2% on average of the cost of food production. Now we're talking about levels from 10 to 20%, and in energy-intensive sectors, even to 60 to 70% of food production. So energy is absolutely important for our sector in order to be able to continue producing food and drink. And of course, all of this has led to enormous cost inflation. So the prices that you're seeing in the supermarkets today are not a joke. And if you look at, for instance, this summer, the consumer prices have gone up approximately by 13% on average in the EU. But producer prices have even gone up further, 19%. So it also shows that we as an industry, but also farmers, other producers, are absorbing some of those costs to try to keep the food affordable. And this is a big, big challenge these days. And then, of course, climate change. We have been talking about that. It's very obvious that we've been facing the largest droughts that we've ever seen in 500 years this summer. But food is the first victim of climate change, and food production is at stake when we are not able to control that. Now, this is leading to cost of living problems. This is leading to production distortions. We saw that when we had a lack of sunflower oil. We have become much too dependent on individual regions or individual geographies for certain inputs. And of course, that is leading also to global problems. So the figure was already given that we are heading towards 10 billion people by 2050. The FAO estimates that we have to increase our food production by 70% from 2005 till 2050. So there's a huge, huge challenge that is coming ahead of us. And if we are not able to tackle that, we risk increased hunger. We're already seeing that in parts of the world, but it can easily also extend to Europe. There are more and more people that have to go to food banks, which is absolutely terrible. And of course also social unrest, what you saw in the Arab Spring. So the impact on the food and drink sector of all of these crises is in terms of the inputs. We need agricultural raw materials that are good in terms of quantity, quality and price. But it's also about access to energy. If we are not able to have gas or electricity, we can't produce, and that's a fact. But also things like packaging have gone up, and this is obviously of concern. But it's also about markets. So when COVID hit, borders were closed, certain markets were no longer accessible. So we had to revert distribution channels. We have seen export restrictions. So often in times of crisis, countries tend to become more protectionist, and that has absolutely been finessed for the supply chains, which are very much global in our sector. And the last point, which has not been really addressed today yet, is labor. We are having a shortage of workers in food and drink sector, but also in the farming sectors. And we need more skilled workforce to deal with the environmental challenges. So in the short term, our sector, and I'm talking about the agri-food ecosystem here, needs lifeline support by being prioritized as an important sector, a sector that is crucial for society and therefore should have first access, or one of the first access to energy. Immediate support, obviously, for the companies that are under bankruptcy. We have in Belgium 4 out of the 10 companies will go bankrupt, they estimate, if another economic shock hits right now. So this is serious stuff, and it is not only in Belgium, it's everywhere in Europe where this is a problem. We need temporary flexibility. Sometimes you need to tweak a bit the rules in order to be able to guarantee continued supply. So whether it's labeling or official controls, imports, a lot can be done, and a lot is done, unfortunately, also by the European Commission. And we need single markets to function because we trade so much. And of course, avoid protectionism. Now, this is a very dense slide, but I think if we have to look at the medium to long term, what is needed is 3 key words. It's investment, it's innovation, and it's partnership. It's investment is needed because we are far from there on the actions that are needed to move towards sustainable and resilient systems. But we need to have a combination of public and private investment. And at the moment, there is no such mechanism that really allows for that. If you look at the farm to fork strategy, it has 0 budget to it. They have the common agricultural policy. Of course, we have other types of funding mechanisms. But if we want to have a sustainable food systems approach, you need to also put investment to it. We need innovation. I'll come back to that. And we also need partnership, because we can't do this alone. We need to work in the supply chain. We need to work with public and private authorities and move on together. So I don't want to go too much into the detail on why food sustainability is important. It's of course a consumer trend, so our companies are seeing that consumers are more and more interested in sustainability, but it's also an expectation that farmers, producers are active and give a positive contribution. It's the regulatory direction of the Green Deal and the Farm to Fork, but it's also a way to build competitiveness and resilience, because the companies that are investing in sustainability are often performing better also on the markets. So how do you approach innovation? We have heard about new genomic techniques as one example, but of course there's much more. And the point I'd like to make is that we need to have a systems approach to innovation. So it is not enough to just look at what we can do to increase the nutrient density of crops, but we need to see how does that work inside the entire supply chain, and what are the demands. And often that starts with the consumer. What is the consumer willing to accept? What is the consumer willing to what is the perception? And we have to work backwards and see how we can then match that to increased food productivity, which is in fact the point that I made before. And that is a challenge, because even if we have great innovations and great technology at our disposal, if the consumer does not accept this, then we still have a long, long way to go. So we have to think about it not just in terms of finding a solution at the farm level, but also looking at what the consumer, how we can convince the consumer to take on these innovations. We have a technology platform called Food for Life, where we are also looking at other types of innovation when it comes to about smart packaging to avoid food waste, when it comes about things like personalized nutrition, trying to see how you can work with consumers in individual profiles and then cascade that back into new technologies and so forth. So there's a lot of innovation potential, but I think it needs to be more systemic. And I think that's something which I also hope that we will discuss in today's conference. Now, a few words on new genomic techniques from our side. The food and drink industry in Europe believes that NGTs can provide valuable potential to improve different traits of crops, plant materials, and ingredients. So we are very much supportive of this. This is part of the toolbox that farmers should have. But of course, it is also true that we have to look at it from a global point of view. If we in Europe don't have access to NGTs, we will lose out on export markets. We will lose even out on our own market here in Europe, because there is a lack of a detection possibility. Consumers need to be informed, but it's easy to resort to labeling approaches there. So we believe that there are different alternatives to putting everything on a label, and there is a lot of work that has to be done on that. Now, we think that, of course, the regulation that is coming, and I'm happy to hear that the Commissioner confirmed that it will come next year. We have to be pushing for that and actively. We hope that it will also apply a differentiated regulatory approach, because what we are seeing and hearing right now, and we wrote a letter with a couple of associations on that, is that the Commission might want to go for a full risk assessment approach to NGTs, which is not what we would think is needed. And I think if SMEs, particularly in our sector, are not convinced that this process is going fast and is going without the administrative burdens around it, they won't be enticed to take up these kind of technologies and these kind of processes. So timing and speedy authorization is really of the essence. Now, concluding, I think it's clear that food needs to be prioritized, and I'm talking about the entire system here, and not just farmers and not just processes, but the entire system, because otherwise we are not able to produce, first of all, but also not to produce more with less and with better stuff. So this is really, really key. If we do it right, security, sustainability, and competitiveness can go hand in hand, but we can't just rely on the ecosystem to work it out on its own. So we really need the public authorities to help us and enable that, with us. And that is calling for the three drivers that make that happen: investment, innovation, and partnership. So if we really get that right, if we lose the momentum right now, we waste 3 more years. And I just want to remind everyone, there are only 7 more harvests until 2030, where a lot of these targets that have been set by the Green Deal and so forth will have to be met. So if we're not having the discussion about investment, co-innovation, partnership now, we will lose out. So we need to have the solutions-driven systems approach to innovation as part of that exercise. And I really hope that we can work that out much more in a coherent way. Lastly, there's no time to waste. So let's start the discussion right now. Thank you very much.
Thank to Mr. Dirk Jacobs for his contribution. Dámy a pánové, teď mi dovolte, abych pozvala posledního přednášejícího v tomto bloku konference, protože i konference je svým způsobem živý organismus, tak se nám malinko proměnil program. Dovolte mi, abych sem na místo původně avizované diskuse nebo odchodu na oběd ještě pozvala k poslednímu příspěvku genetika pana profesora Aleše Pečinku z Ústavu experimentální botaniky a Akademie věd České republiky. Pan profesor Pečinka je rostlinným genetikem, který se zaměřuje na analýzu DNA a její regulaci v Centru pro strukturní a funkční genomiky rostlin Ústavu experimentální botaniky. On svůj doktorandský výzkum prováděl například na Leibnizově ústavu Plant Genetics and Crop Plant Research v Gletterslebenu v Německu, působil i na institutu Gregora Mendela ve Vídni a jako vedoucí skupiny v ústavu Maxe Plancka pro výzkum šlechtění rostlin v německém Kolíně nad Rýnem. Tak to je jenom stručné představení, pane profesore.
Tak, děkuji za úvod. Hello, dobrý den. It's a great honor for me to be here today, and my talk will be a little bit more scientific than the talks of the previous speakers, and I thought, I will look a little bit away for the moment from the complex political, legal, and regulatory issues we are discussing, which are very important, and I would like to discuss with you or show you by the eyes of scientists why I think that new genomic techniques are really scientific discovery of the century, and why they could be very helpful and essential in future plant breeding. And yes, there is already my talk. So I'm going to tell you some of the innovations where I think the new breeding techniques could be very useful in solving long-term problems, and also would like to show you what we have actually in our scientific toolbox, what we can use and how these things work a little bit. So plant breeding has always used let's say, human knowledge, and as the human knowledge grew in the time, we were using for breeding the latest available techniques. And in the Middle Ages or in the prehistory, it was simple selection of plants which appeared randomly in the field or even before in the nature. After understanding the principles of inheritance, which were laid actually here in Czech Republic by the Gregor Mendel in the 19th century, then we could apply the laws of genetics basically to make the breeding more efficient and to speed it up and make it more directed. And in the 20th century, our toolbox grew up very much for lots of other techniques, including mutagenesis, which led into very intense mutation breeding. And I would like to stress out that thousands of varieties we are currently using in our fields are based on mutation breeding. Then the techniques of molecular cloning, in vitro techniques, transformation, in the last decades also genomics. And since, let's say, 2 decades, this toolbox grew also for the techniques of genome editing. So if we look on these genome editing tools, sometimes they are called as molecular scissors, or we could maybe call them also as a molecular scalpel, because I think the term genomic surgery is very, very nice, very well fitting. We have actually several types of those molecular scissors or scalpels in the toolbox. The first ones which were discovered were so-called zinc finger nucleases and tail ends. And these techniques, they use a specific protein part, which you can see here. And this protein part, that's the locator. This can bind to specific DNA sequence, and this provides a location for the nuclease. And to this protein sequence, there is attached a nuclease, in this case, which is called FOC1. And this nuclease, because DNA has 2 strands, it's like a little bit like, if we show it simple, like railway line. Each of those nucleases will cut one of those strands. The latest technique which basically appeared in this toolbox is a method of CRISPR-Cas9, and as mentioned, that's technique which is used by bacteria for protection of their genome against genomic invaders such as viruses. And this technique is using short RNA molecule, which is making complex with protein Cas9, and together this complex will find target sequence and will simply cut the DNA. And this was really key finding, which was awarded with the Nobel Prize in Chemistry in 2022 to 2 scientists, Emmanuel Charpentier and Jennifer Doudna. So these techniques, they really allow lots of modifications, lots of changes. So one of them is genome editing, but I will also shortly mention localization. So we can have 2 basic variants if we talk about Cas9, Cas nuclease, which can cut both DNA strands, and Cas nicase, which can cut only one end. And we can have simply just a small change, so a cut which results in a small insertion or deletion of sequence, which would call into so-called SDN1 techniques. And then we can have, we can provide donor DNA, and then depending on the type of mutation, whether this donor DNA again introduces just small change, we would call it technique 2. And if the donor DNA contains even some kind of foreign DNA sequence and just the ends are homologous to the target sequence, then we would call it site-directed nuclease technique 3. So these techniques, all of them can be used, and depending on what scientists need. In case we mutate those scissors or the scalpels which are in this Cas9 enzyme, we still keep its molecular function as some kind of like a messenger. It will find the location but will not be able to cut anymore. And with this we can modify locally molecular properties at that genomic position. So I would like to show you some examples just briefly, because at least some of them were mentioned already before. So with simple cutting of the genome at specific position, we can speed up breeding. And there is tens or hundreds of examples in the meantime, such as modified tomatoes, which require much less space. Already mentioned wheat, which does not germinate in case the harvest falls into the rainy season. Tomatoes, which could provide provitamin D to people who simply, for some reason, do not have enough sunlight or do not want to eat fish products. Oops, sorry. And all kinds of resistances, even in the very complex genomes. Sometimes what we hear with those techniques is, yes, but how do you know that you are not introducing mutations somewhere else in the genome, that your system is specific? And this is really the power of this genome editing, because if we look on what happens if we use the nuclease, which has the 2 scissors, versus the nickase, which can cut only one strand of DNA, you can see that here at the position where the nuclease was targeted, there is lots of mutations. This is fine, this is what was wanted, but if you use nickase, nothing happens. Even when you target it there, nothing happens. The nickase alone cannot modify the genome. And it means it cannot also introduce mutations somewhere else. But if you combine two nickases, you put them close, you target them to close positions, you suddenly see appearance, lots of insertions and deletions. So we can really increase the specificity, if needed, of the system by at least one order of magnitude. And as already mentioned, if we use mutated nuclease without possibility to cut, we can send some modifier protein, we can combine it with a specific domain which will not change the DNA sequence, but will change so-called epigenome, the proteins which are binding to DNA. And with this, we can really modify even a very important agronomic trait such as flowering time, here in this case, which is based on zinc finger nuclease, which was using human DNA, so-called demethylase. You can see that this reprogramming led to change in the flowering time in one very important gene in the model plant we are using. So it's really amazing what these techniques can do. And what we know is, what was already mentioned, the classical breeding really gave us or used or we had the Green Revolution, which really allowed enormous growth of human population and increasing the standards of living. However, this is connected with very high costs, which were mentioned by many previous speakers. So I would like to mention in the second half of my talk some of the problems which classical breeding is facing and how these new genomic techniques could help there. One of the arguments is that with these new genomic techniques, we could really make much lower number of mutations in the genome, so we could keep mutations under control. As already mentioned, thousands of varieties are based on so-called mutation breeding. And the mutation breeding looks like this. You take seeds or some part of the plant, you expose it to chemical which is making lots of mutations or some kind of gamma radiation or X-rays, and the mutations are created randomly in the genome, and then you select a lot, the one which you need. So then this is a real example of chemical mutagen in the plant genome, and you can see positions. So these black bars represent individual mutations, and there is more than 1,000 all over them in the plant genome. However, with these new breeding techniques, we could really target the nuclease to one position and create one specific mutation and keep the rest of the genome very clean. That means this is very much in the spirit of the genome surgery. The classical breeding, as applied now, is very much dependent on plant hybridization, and this is really great tool which gave us already Gregor Mendel and explained us how to use it, and we are using it until now. However, the breeding based by hybridization means that very often you destroy the good combinations of genes which you created, and this takes years and lots of efforts and lots of resources. Imagine that you have elite cultivar which is having high yield but is maybe sensitive to some kind of pathogen, and then you have landrace which has low yield and is highly resistant to this pathogen. If you create them in the first generation, you have half of the chromosomes for each chromosome, one from one parent, another one from the other parent, and then these chromosomes start mixing, and then you really end up with some kind of mix which may have some good traits, some bad traits, and it takes very long time and lots of generations to get what you actually want. With genome editing and the knowledge of basic research, you can just use the elite cultivar, modify one gene if you know what you need to modify. There's of course this knowledge required at the beginning of these techniques, and then you can end up just with 2 generations with the uncompromised genome of the elite parent and with the desired modification. Sometimes in the classical breeding, you also can have this pathogen resistance. You know, you've been selecting for your 5 generations. You have pathogen resistance introduced, very nice, but you appear to be in the bad neighborhood. You may have gene which is responsible for bitter taste, for example. And how do you get rid of it? How do you remove it? You have to make more screening. You go through thousands of plants more, you make more generations, and if you are lucky, you really find a combination where you have pathogen resistance and the bitter taste or the unwanted trait is gone. However, that does not work always. So again, with genome editing, even with such line, one can focus on the bitter taste gene, edit it, and just get the desired combination of traits. We already heard that healthy food is one of the main focuses of the food production and agriculture in Europe, and we know that some, let's say, traits which are difficult, which are connected with gluten intolerance, with celiac disease, are very difficult to tackle with a classical breeding. This is because in bread wheat, the gliadin genes, especially alpha gliadins, which are responsible for the gluten intolerance, are occurring in many copies. You see there is about 30 copies, and removing or modifying all of them by classical breeding would take enormous amount of efforts and time. So here, the genome editing offers one possibility is, of course, mutating some of them to reduce the gluten load, but they could also be edited, you know, in order to reduce immunogenic potential of those gliadins. And finally, as already mentioned, we can have de novo domestication for stronger crops. Many of our crops, as they were selected in the process of breeding, the prime trait was just yield. However, with this they lost very often some kind of resistance to different pathogens or some other traits. And we could use the techniques of de novo domestication. I think so far the best demonstrated in tomato or in rice. And just with a few, you know, generations, with modifying a relatively low number of genes, we can get de novo domesticated crops, which have some of the beneficial traits which were lost in the process of conventional breeding. And this may not be so much of interest here in Europe, but in some parts of the world, especially in Africa, the classical domestication is not that far, and this could be very interesting to extend the portfolio of agricultural plants, of crops in those regions. So I'm at the end of my talk, so I hope that I convinced you at least a little bit today that genome techniques could be very useful addition to what we have already. And in some cases where basically the classical breeding is too slow or too expensive or would require too much resources, these techniques could be really very useful. They could kick in and could provide some solutions which would make our breeding faster, cheaper, and more efficient. So with this, I would like to thank you for your attention, and I think I wish you good lunch.
On už to, pan profesor to vzal za mě, v podstatě to moderování, protože už vám popřál dobrou chuť k obědu. Já ještě jednou děkuji panu profesorovi, držím palce vašemu úsilí, zejména co se týká toho, abyste dokázali vypěstovat, nebo aby zemědělci dokázali vypěstovat třeba v Brzku nějaký druh obilí bez toho lepku, který je tak zákeřný pro všechny celiaky. Tak to je jenom takový můj soukromý postřeh. Dámy a pánové, pokud se podíváte na hodinky, tak my jsme v lehkém časovém skluzu, nedá se nic dělat. Toho prostoru na oběd bohužel nemáme víc. Tento dopolední blok uzavíráme, před námi je ještě odpolední část konference. Pojďme si říci, že se tady opět setkáme ve 14 hodin 30 minut. Tak já se na vás na všechny budu těšit. potěšit, protože nás čekají další velmi zajímavé příspěvky. Byla by škoda je odsouvat dál. Čeká nás také velká panelová diskuse. Pochopitelně při ní budete mít všichni prostor se našich vzácných hostů ptát. Jenom teď mám takovou malou technickou poznámku pro všechny kolegy z médií, pro novináře. Pokud by se chtěli našich hostů, kteří stále ještě zůstávají účastníky konference, na cokoliv zeptat, tak ten prostor nastává právě v následujících minutách, kdy vám hosté budou k dispozici. Ptejte se, na co budete potřebovat. Ano, Zuzko? Bezvadné, takže nová informace, že se tady setkáme v sále ve 14 hodin 50 minut, tak ten čas na oběd je přeci jenom klidnější. Dámy a pánové, já vám všem děkuji za pozornost. Za 10 minut 3 opět v sále. Na viděnou.